Welcome to Click for IB Biology. We're studying genetics, and in this part of the course, what we're looking at is the type of cell division called meiosis, the phenomena called crossover, and how it adds to the variation in the genetic variation in a population. This particular video is aimed at students at the standard level. For students doing higher level, crossover is visited again when we're looking at linked genes. So that's a later video to come here. However, this does introduce the idea of increased variation. There's a blank version of this presentation down in the description, of course. We have 10 questions. There are answers, and you can do those to consolidate your understanding of the content that we're covering here. So the Mendelian laws, the Mendelian laws of segregation and independent assortment, had been published by the beginning of the 20th century, and geneticists were carrying out tests and exploring those concepts and consolidating the evidence to support Mendel's assertions that he had published. However, around 1909, Jansen, through his microscopic observation of chromosomes, observed this phenomena here. And here we have this is a homologous pair of chromosomes. And if I just draw over the top to make it simpler, so there's a chromatid and here's a chromatid. And then on the other side, there's a chromatid, and here's the other chromatid. And what he noticed was that along the alignment of these pairs, these homologous pairs, if I just draw this on the right-hand side, just to perhaps simplify the whole thing. So at prophase 1 of meiosis, we have the alignment of the homologous pairs. And the adjacent homologous pairs come very close to each other in a process called synapsis. So this is what Jansen was actually observing. And he noticed that there seemed to be a crossing over of, I'll just put dots to show which are the chromosomes. So these two chromosomes here, these chromatids rather, are overlapping, and we can see that in Jansen's photographs. And here they're illustrated at this point here. And such crossover points are called a chiasma. And what Jansen appeared to be showing was that the chromosomes were exchanging lengths of DNA. Just a year or so later, in studies on fruit fly breeding behavior, Morgan Hunt started to observe outcomes in his genetics crosses on the fruit flies which were not predicted by the Mendelian laws. So at this time, they're consolidating information about Mendel's rules, and yet items arose in Morgan's genetics experiments and from Jansen's study of chromosomes, which appeared to contradict what Mendel had proposed. This phenomena is still called crossover, and in this process, lengths of chromatids will exchange. And so if I just do a brief illustration of that, like so, and you see this crossover point here, and so there's an exchange of pieces of DNA, lengths of DNA, and most importantly, the alleles which occur on them. You can see in this image here that there are multiple crossover points along these chromosomes in synapsis of prophase 1. Hunt was eventually awarded the Nobel Prize in Physiology and Medicine in 1933 as he had pointed out, it wasn't appreciated at 1911, but this points out where a huge amount of the genetic variation in a population has its origins in the process of chiasma. Multiple chiasma are called chiasmata. So just to kind of give you a taste of what's going on, the Mendelian laws were predicted like this, that we have allele big A here, and that's linked to allele big B. And so wherever the big A allele goes, the big B allele should go. And that argument is sustained on the homologous partner. Wherever little a goes, little b should go as well. And so the prediction was very much like this, that through the rules of meiosis 1, we separate the homologous pairs, and in meiosis 2, we separate the chromatids, and therefore 50% of the gametes should look like this, and 50% of the gametes should look like that. But that's not what Morgan observed. What Morgan observed was that these ones were in high frequency. This one as well was in high frequency, so we might call those the normal ones. But then we had this exchanging process at the chiasma, which created these unusual combination of linked alleles. Now, these combinations in the gametic cells were at low frequency. So this is the main point which emerges from this particular feature, is that whilst we've already learned that meiosis is responsible for tremendous genetic variation, there is even more genetic variation available when we take into account crossover. And that's the significant learning point of this particular video. Okay, we're moving on in our studies from genetic variation. Here's the next video link. There's the chance to subscribe to the channel, and of course the playlist with the entire genetics videos list.